All right, so I'm David Anderson. I'm the director of the Chen Chao and Chrissy Chin Institute for Neuroscience at California Institute of Technology. Have been that since 2016. And it's a pleasure to be here and to chair this session on what overall looks like applications of AI to medicine, although there isn't an official title for this. It's just called Session 2. So we have 3 speakers today. I've asked each of them to keep their remarks to 25 minutes, so we have 5 minutes for discussion, and I'll introduce them briefly. Let's get started with Mariam Shanetchi. Did I pronounce that right? Mariam is the Alexander Sawchuk Chair in Electrical and Computer Engineering and a professor of Electrical and Computer Engineering, Biomedical Engineering, and CS at USC, CSUSC. She trained at MIT, where she got her PhD, and she does research at the intersection of engineering, AI, and neuroscience to develop closed-loop neurotechnology and brain-computer interfaces. She's had a number of important awards, including ASEE's News SN10 and many other Young Investigator Awards. So welcome, Mariam, and we look forward to hearing from you.
Well, thank you very much, David, for the introduction. It's a pleasure to be here today and talk about some of the work that we've been doing on developing AI-driven neurotechnologies. So as you may know, brain disorders are actually a leading cause of disability worldwide. Actually, 1 in 5 US adults suffer from mental health conditions. Now, unfortunately, our treatments do not work for a lot of these patients. For example, 20 to 30% in depression are treatment resistant. So the question is, what do we do for these patients? Now, based on the understanding that these disorders are actually signs of abnormal activity patterns across a network of brain regions, one idea we're pursuing in my lab is to try to directly decode and regulate these abnormal activity patterns towards potentially transformative therapies. So what do these activity patterns that I'm talking about actually look like? I'm showing you here intracranial electrodes that are implanted in the human brain, and these are signals that we record from them. So you can see that these are very complex, high-dimensional, noisy spatial-temporal patterns. So what we envision in my lab is to build a closed-loop system that models this complex activity patterns, decodes from them an underlying state of the brain, let's say mood in depression, and then uses that as feedback within a controller that would then adjust the therapy parameters, let's say neuromodulation, frequency, and amplitude, in order to take the brain to a target healthy state. Now, this is an example of a closed-loop control system. And as engineers, we're actually very good at designing these systems. Think about robotics, think about autopilots. But unfortunately, we still cannot do this for the brain. And you may ask me, why is that? So of course, the problem here is that we didn't design the brain. We don't have those beautiful equations for it. So this is an unknown system with over 80 billion neurons, and this introduces a huge AI challenge to build data-driven models of this complex system and to come up with data collection frameworks that can enable this modeling. And that is precisely what my lab works on. We work at the interface of control theory, AI, and neuroscience to develop next-generation BCIs and closed-loop neuromodulation systems that can restore movement, for example, in paralysis, but also go beyond that and think about restoring lost emotional function in neuropsychiatric and mental health conditions. So what are some of the challenges we have to overcome to build this system? The first challenge, of course, is to build this decoder. Now, when we started a few years back on this problem, decoding of mood and mental state had not been demonstrated. So to solve it, we first started by collecting the right data sets. In collaboration with Eddie Chang, who's a neurosurgeon at UCSF, we collected intracranial EEG from epilepsy patients over several days. And then while these patients were in the hospital, we asked them to intermittently report their mood using an iPad-based questionnaire a couple times a day. So this was the data set we had to use to train our models. Now, this is, of course, a difficult machine learning challenge because we have a very high-dimensional neural signal space to model, but very few mood labels to model it with. So back in 2018, we actually showed that we can solve this problem and achieve the first decoding of mood from human brain activity. And we did this by essentially developing a dimensionality reduction for neural data. First, our method identified smaller subnetworks that were good enough for decoding. And then even within those smaller subnetworks, we further reduced dimensionality by training a latent state dynamical system model. And then we took these low-dimensional states and just related those to our sparse mood labels. This actually allowed us to decode mood variations in every single individual. So this was very exciting. Now you may ask me, okay, great, but what were these subnetworks? So we found that even though we were building our model in an individualized way, across patients, the decoder picked consistent limbic regions as being mood predictive. But even more interesting than that, in 60% of patients, this area of the brain called orbitofrontal cortex was picked as the most predictive site. Now, why is this interesting? Because in collaboration with Eddie, we went ahead and actually stimulated there. And what we found was that depression symptoms were actually alleviated. So patients went from describing their feelings to be on the sad side or a little bit nervous to describing it as being calm, cool, and collected and feeling a lot better. So these two different studies, one from a machine learning angle, one based on neurophysiology and simulation, arrived at this common node, orbitofrontal cortex, and showed its significance in the emotion regulation network. So this is great. This showed we can directly decode mood from the human brain, and it opened the door to personalized therapies for mental health conditions. But unfortunately, in order to make these systems translate, we have to significantly improve their precision and robustness. And the way we're doing that right now is by tapping into the power of AI to develop foundation models for the brain. So you might ask me, okay, why foundation models? How's that going to help you? So the first challenge we have in these cases is that we actually have very limited data in each individual. So if I build a foundation model, I can actually pre-train it by pooling data across many individuals and allow it to actually generalize. The other challenge we have is that we have very limited behavioral labels per individual. For example, I showed you those mood reports. There were very few. But a foundation model can be pre-trained completely agnostic to behavior. And again, pool labels across individuals to generalize across different behavioral tasks. So the idea is if I pre-train this model, I already have a good model, a new subject comes in, it has limited data, fine. I can just simply fine-tune this model for that subject and have a model that's already accurate. Now, of course, this probably reminds you of what large language models did for language data. But we have very distinct challenges in this case. First of all, if you think about these electrodes, they're very spatially sparse and heterogeneous from subject to subject. They also involve very complex spatio-temporal interactions. And indeed, how we should encode spatial information in neural foundation models is currently an open question, with prior neural foundation models either not encoding space at all or encoding it at the level of single channel coordinates. But there are other ways we can encode space. For example, we can encode which brain region an electrode belongs to. We can even go larger and encode which brain lobe the electrode belongs to. So what spatial scale should we encode? This remains unanswered. So with this motivation, in a paper that's upcoming, we developed a method we call Barista that flexibly incorporates and tests for the spatial scale within neural foundation models, whether at the level of single electrode coordinates, whether at the level of brain regions or atlas parcels, or even at the larger scale of brain lobes. And Barista does this during the process of tokenization by taking these neural activity patterns and encoding not only temporal structure, but also spatial designation of those electrodes to get latent neural tokens that are then input into a transformer. Then we pre-train this entire model fully agnostic to behavior with a self-supervised mass reconstruction loss. Once we do this, then downstream we can train a simple decoder to decode various behavioral tasks. So what do we find? We, as a first proof of concept, tested this model on a benchmark data set called Braintree Bank, in which 10 patients were watching movies. And we took the neural activity, and we wanted to decode from that various auditory and visual features in these movies. For example, whether there was speech or no speech within a given neural data segment. So what do we find? First, well, we find that indeed pre-training by aggregating data across subjects does much better than single subject models, showing the advantage of this foundation modeling. More interesting than that, we find that by encoding spatial information at the level of brain regions, Barista significantly outperforms state-of-the-art neural foundation models. This also extended to other downstream tasks, for example, sentence onset decoding. So what this shows us is really important to be careful about how we encode space within the brain in these neural foundation models. The other interesting part was that even though we were encoding space at the level of brain regions, we could still reconstruct channel signals almost as well. Further, Barista generalized to new subjects that were completely held out of pre-training. And finally, it scaled, its performance got better with more and more pre-training data. So we're very excited about this because we think it's getting us closer to decoding methods that are really robust and precise for translation. But unfortunately, that's not enough. Because even if I can decode somebody's mood perfectly, I still don't know how to stimulate their brain to regulate or change it. So what we need additionally are input-output models that describe the effect of neuromodulation and stimulation on brain network activity and therefore on the underlying mental states. So towards this goal, we've developed data-driven input-output dynamical models. And we did this by essentially incorporating the effect of stimulation inputs within the same low-dimensional representations. So this is fine, but the main challenge here is how do we actually collect informative data to train this model? In particular, we have to stimulate the brain somehow, record the response, get the data set. But how do we stimulate the brain to get informative data? So towards this goal, we've been developing novel neurostimulation patterns that start with a basic pulse that's used in the clinic, but stochastically change its frequency and amplitude, essentially according to a coin flip. Doing this, we get what we call a binary noise modulated electrical stimulation pattern that we show can persistently excite the brain activity across a range of frequencies such that we can learn accurate models. So once we confirmed this, when we started a collaboration with a non-human primate neurophysiologist, Bijan Pesaran, and actually applied this waveform in the brain. And what we found was that indeed our models could predict and forecast how neural activity changes in response to a time-varying neuromodulation. So this is great. But one thing you may notice is that I've kept this model so far linear. And there's a very good reason for that. It's because these linear models are actually great for building closed-loop real-time systems. And that is due to their ability for flexible inference, meaning the same model allows you to do real-time inference or filtering, non-causal inference or smoothing, or even inference in the presence of missing neural samples that can happen, for example, in wireless PCIs. But there's, of course, nonlinearity in the brain. So our next step was to develop AI architectures that not only capture nonlinearity, but also enable flexible inference. And we call this architecture defined for dynamical flexible inference of nonlinear embeddings. So the intuition behind define is actually pretty simple. What if we capture the nonlinearity within a nonlinear manifold embedding? For example, this torus, and then capture how neural activity moves on top of that according to a linear dynamical system model so that we can do a flexible inference. Essentially, that's what define does. So this is the generative model. We have a manifold latent factor layer that we non-linearly relate to neural data according to a decoder network. Then on top of this, we write a linear dynamical system model for the evolution. We can also incorporate neuromodulation inputs in this model. And then we fit this model completely end-to-end to forecast neural activity. Now, how does this allow for flexible inference? I can encode the manifold factor. It's the output of a linear dynamical system model, so I run my favorite Kalman filter. It's flexible. How does this work? So we tested this on a reach and grasp non-human primate data set in which animals were reaching and grasping to distinct locations. And the idea was to decode their movement from their neural activity. So first we confirmed that yes, define can do this in real time, even when you had missing neural data. It was more accurate than the linear model, as well as benchmark sequential autoencoders. And interesting, it was also very robust to missing data. So you can see the degradation in define is much lower. So this was great. This shows you how this architecture for BCIs and neuromodulation systems. But I have to still incorporate the effect of neuromodulation. So since then, we have actually built a variant of define that can model input effects. In this case, we tested this for modeling the effect of deep brain stimulation on calcium imaging data. So here I'm showing you define can forecast the calcium response to DBS and do so more accurately than linear models. Okay. So that's how we are addressing the second challenge. Now, there's also another problem that we have to be very mindful of when designing these systems. And that is the fact that the neural activity we record does not just relate to the mental or behavioral state that we are interested in. For example, like right now, I'm talking to you, I'm moving my arm, maybe I'm hungry. All of those states simultaneously are represented in my brain. So the issue is that it may be that just a minority of brain states that give rise to your neural data may actually be related to your behavior of interest, essentially those green behaviorally relevant states. So how do we disentangle just those so we can modulate those and leave everything else intact? This has been a motivation for several years for us. So to do this, we've developed a novel framework for jointly modeling neural and behavioral data, starting with a learning algorithm PSID back in 2020 that did this within a linear dynamical system modeling framework by developing novel subspace identification methods. So these were analytical methods, actually. So how does this work? Let's see, get an idea. Let's go to my favorite data set, the reach and grasp data set. We were again decoding movements using PSID and using an unsupervised model. And you can see that regardless of what latent state dimension I use, PSID decodes movements better. Okay, great. But more interesting than that, PSID could do this using a much lower dimensional state, meaning 5 times lower in this case. And that is exactly because of this new capability to just disentangle those parts that you care about and get them more accurately and robustly. And this held even when we compare to nonlinear unsupervised methods. Now, we've extended PSID in multiple ways since then for inter-regional communication discovery, for generalized linear models, and so on. But at the end of the day, the backbone of PSID is a linear model. So obviously, our next step was to go nonlinear. So we developed a deep learning architecture recently that we call DPAD, for Dissociative Prioritized Analysis of Dynamics. So DPAD writes this relationship between neural activity and behavior in terms of 4 interpretable mappings: a mapping from neural activity to states, the dynamics of those states, and then the mapping back to behavior and to neural activity. So it's interpretable to some extent. And then we make each of these mappings a nonlinear feed-forward neural networks. We allow for categorical behaviors, such as cognitive states. We allow also for intermittent behaviors. And then we allow the model to run on naturalistic data without any trial structure. So how do we do all of this? Of course, let's just give you an intuition. Essentially, DPAD is a multi-section recurrent neural network model that we train jointly with neural behavioral data. In the first section, we tell DPAD, hey, just get me the behaviorally relevant state, and do so by predicting future behavior from past neural activity. Once we get those states, then we tell, okay, how much of the neural dynamics have we already not explained? Find those residuals and fit the behaviorally irrelevant dynamics. So that's how we get this disentanglement. So how does this work? We compare DPAD across many datasets with state-of-the-art methods and show that it outperforms them both in terms of neural prediction as well as behavior decoding. So this is the reach and grasp dataset, and these are 4 other movement-related datasets that we looked at. So this is great. So far, we have our neural behavioral model, but we still don't have the effect of input in this model, whether it be stimulation, sensory, task instructions, or even an upstream brain region's activity. So in our recent work, we've actually extended these to also incorporate the effect of input in these neural behavioral models, such that we can disentangle the behaviorally relevant dynamics, not only from the irrelevant stuff, but also from input dynamics, so that we can look at intrinsic neural computations. Now, we've used these disentangled representations to look at an important basic science question, and that is whether intrinsic neural dynamics and computations are similar or distinct when animals perform different tasks, or from animal to animal. So why is this hard? This is because these tasks actually have different inputs. So if you think about inputs, these are visual inputs in terms of, okay, which target do you have to go to? And when you change task, you change input. And if you don't take into account differences in input, you may actually be misled into thinking intrinsic dynamics are different. But that's not the case. It's really inputs that were different. So because we can disentangle the input effect, we looked at this question. First, we used an unsupervised version of our model and looked at overall intrinsic dynamics in the motor cortex. These are dynamical modes that we looked at. And you can see that these pictures look very different. So great. So overall intrinsics are different. Then we said, OK, let me just zoom in on the behaviorally relevant subtype of these dynamics, those that are related to execution of movements with our method. And you can see that they're remarkably similar across animals and across tasks, unlike the intrinsic overall dynamics. So this shows you that there are these preserved neural computations between animals that are involved in the executions of movements. So we built our decoder. We built our input-output model. The next challenge, of course, is to build the controller. And that is a huge challenge that we're currently working on. We are also working on making these systems multimodal, whether it's different modalities of electrophysiology, whether it's neuroimaging, optical, acoustic, magnetic, or even wearable data or physiological data. We want to fuse information across all these modalities to get the best accurate readout of mental states. We're also interested in different modalities of neuromodulation and inputs, whether it's electrical, optogenetic, magnetic, focused ultrasound, or even pharmacological intervention. So far, we've done this multi-scale modeling for different modalities of electrophysiology. Spikes at the level of single neurons, local field potentials at the level of larger populations, and then intracranial EEG. So in a recent paper, again, that's coming out in a month, what we show is that we can actually build multimodal models that describe how a brain state simultaneously is encoded in LFPs and spikes, and we can fuse information across these modalities, in this case for decoding of movement. And if you compare the decoding of this movement using spikes with when we fuse modalities, you can see that we can actually get much more accurate. We've also extended these to neuroimaging, including functional ultrasound imaging as well as wide field calcium imaging. Now, for images, a major challenge is that you have complicated local and global spatial patterns. So to solve this, we developed a network architecture that gets the local spatial patterns with convolutions and gets the global one with self-attention mechanisms. And we showed that by doing this, we can significantly reduce the decoding error of behavior compared to all alternative methods. So this is where we're going, a multimodal closed-loop BCI and neuromodulation system. And because this system is going to be multimodal, we're hoping that it can seamlessly extend across a variety of hardware devices that are being developed today, as well as a variety of brain conditions. Whether you're talking about mood disorders, or chronic pain, or obsessive compulsive disorder, or movement disorder and paralysis. So at the end of the day, our vision is to develop an AI-based system that not only extracts mental states from human brain networks, but actually goes beyond that and regulates these states towards potentially transformative therapies for people with neurological and neuropsychiatric disorders. So with that, I'm just at time. I would like to thank my team, as well as the funding sources. I'll be happy to take any questions.
Thank you, Miriam. Excellent talk.
Yeah.
Any questions? Come up to the microphone. Or
All right. Thank you so much for that excellent talk. It covered a lot of really intriguing directions of research. If you could just go back to the previous slide. So one thing that I was kind of wondering throughout the talk is, like, I recognize you're doing this, extracting mental states and regulating them, developing the technology to do that for, like, noble ends, for disease relief and so on and so forth. But, like, this seems very potentially dual-use sort of technology. There are a lot of people who would be very interested in extracting mental states from human brain networks and regulating these states for reasons that I think we would not approve of. And I kind of worry we're in a similar situation with regards to, like, mass surveillance technology, where sort of it was unthinkable to do this on mass scale for bad reasons before it was technically possible to do it. But once it became technically possible, we're suddenly living in a world where it's, like, ubiquitous. So I was wondering about your opinion on those sorts of risks, how we might address them preemptively, and like that sort of thing.
It's a fantastic question, and I get that a lot, actually. So obviously this is for patients who really have explored every other option and nothing works for them. So these are for severe cases. Now, of course, as with any technology, there's a chance that it would be misused. Right? This is not specific to BCIs. But I would argue that we not doing this is almost unethical, because there are people who are at suicide risk. There are 20 to 30%, 5 million in the US, who suffer from major depression and don't respond. So we have to do this, but we have to put guardrails. And for that reason, we work with neuroethicists to think about issues such as neuroprivacy, agency, how people can have a say in how they want their brain to be regulated. So we're hoping by doing that, we can avoid those risks.
quick question, then we have to move on.
Thanks. So really nice talk as always, Mary.
Thank you.
Something that I find in my data is that if you just look at the basic biomarkers that you're measuring, or whatever extracts we're getting from the voltage, those vary over timescales related to attention and arousal. On timescales, they're much shorter than changes in mood.
Yes.
How is it that you think that the decoders are able to capture that in, presumably in the latent space?
Yes.
Do you think, what is it you think that's happening?
Yes.
Because that's what I struggle with, right?
Yes,
And I always
yes.
try to sort of linearize the biomarkers before performing
So that's a
classification.
great question. So I think one of the areas that I think directly speaks to what you're worried about is the disentanglement direction, right? So as I mentioned to you, these brain activity patterns to relate to a lot of things, as you beautifully said, attention, arousal, mood. So what we try to do is by using those mood labels and supervision and going to a low-dimensional latent state representation to disentangle just those neural patterns that relate to mood by finding the shared dynamics between the two in the training data. But you're absolutely right. We have to do that disentanglement, otherwise it could be a needle in a haystack problem. And the reason we can do this is because we have labels and we have a lot of data and we have gotten a good representation. But we can chat more if you're
Okay, we're going to have to move on.
Yes.
Let's thank Mariam
Okay.
again for a great talk. Okay, the next speaker is Jiangdong Liu. He's Associate Professor of Neurology at Baylor College of Medicine, and he is going to be talking on empowering clinicians with AI, a new era of collaboration for better patient care. Please welcome Jiangdong.
All right. So the first speaker talked about Alzheimer's and Parkinson's and OCDs, which are oftentimes considered a higher prevalence and common disease. We're going to switch gear and probably talk about rare diseases. And those are individually rare, oftentimes have a prevalence rate of 1 out of 20,000 or 1 out of 5,000, depends on whether you're in European or US. And then, but collectively, there are about 7,000 rare diseases. And in the US alone, we have probably 25 million to 30 million patients living with a rare disease. And about 30% of them are diagnosed with genetic tools, and 70% of them are not diagnosed or not diagnosable. So the key question here that our institute is trying to address is how to solve the unsolvable. And oftentimes it takes a lot of efforts for these patients to go from clinic to clinic until they reach the right specialty, to reach the sequencing centers to get their genome sequence, get the right kind of specialist to analyze and identify it. And odyssey time is oftentimes 6 years. And noticing this problem, which is particularly severe in neurological diseases, Texas Children's has established an institute called Neurological Research Institute to primarily focus on this category diseases. And we opened our door around 2010, and we've been working kind of closely with hospitals, with patients, families, to gather the data and to kind of pass through, identify the pain point of this process and trying to streamline the entire process. So since 2016, the entire team, this is really not just my lab's work, the entire institute worked together with many physicians and also basic science researchers. We have identified the 71 new disease genes, as you can listed here. The point is not to kind of one by one to understand what they are, but really there is a system and a pipeline that we established can enable a repeatable success in solving these unsolvable cases previously. And the core part is basically this flying wheel that has 4 components, starting with patients. And we collect very detailed phenotype information from the patients at clinic, and then their data will be passed to the computational center using bioinformatics tools, and more recently AI tools, to narrow down the search space to figure out what are the potential candidates we can evaluate. And those short list candidates will be moved to the model organism core. In this case, we use the fly as a kind of an intermediate organism to further narrow down the list. Along with other human genetic tools, we can identify and diagnose these cases. And then it will be pushed to another team in the institute for potential drug and therapy development, and it will be pushed back to clinical trial, and again, kind of back to the hospital. I'm going to dissect this process and trying to illustrate how computational tools and AI is involved in every step. Of course, this is a very complicated process, and I'll highlight things that I'm involved and our team's involved. The first part is how can you narrow it down when a person is sequenced? Oftentimes you're dealing with hundreds of thousands of variants in our genome, and how do you quickly narrow it down to a handful target that you can move on for experimental validation? Well, there are many solved cases, probably a couple thousand solved cases within our sequencing centers. Initially, when we worked with clinical geneticists, the way to do that is really to kind of have a little bit script here and there using handcrafted rules to filter down the list, and then manually review every evidence across multiple databases. There are about 150 features that they have to query across various data sources, and then they combine and integrate all these features together into a ranking metric to shortlist it. So the first thing we did is actually to collect all the data, construct the handcrafted feature space, use the label that has been solved, and then put it on a very classic machine learning framework with training, testing, cross-validation, and find some sort of a classifier or ranking algorithm to do it. And this is a kind of very straightforward, but the getting the data has a lot. Like most of the healthcare problem, getting the data, getting the permission to use the data takes much longer than actually building the algorithm. And the algorithm was kind of tested and then deployed to the clinical side, and we got feedback and then benchmarked it and published it. So here you can see, some of the performances metrics and benchmark against existing tools. It seems to have significant improvement in terms of accuracy. And then later on, we kind of realized that the one challenge that when we communicate with our clinical geneticists is not interpretable. Even when you have an XG boosting algorithm, and then there are ways to extract what feature is important, but it's not really flexible to communicate with a clinician and why this case is solved based on what evidence. And that's where we put on, using these large language models on a much smaller list. We tried to put this on a much longer list, like hundreds of thousands of variants, but it's just very slow, and we can't really, within the reasonable amount of time, to get it finished. However, we can always do this on a much more manageable list that's shortlisted by the first machine learning algorithm, and then run a follow-up foundation model to re-rank the candidates. And it turns out this will give much more explainable result, and also it slightly improved the accuracy of the previous existing algorithm. As you can see in the blue curve here, compared to the gray curve, it has a slight improvement in terms of accuracy. I'm going to walk through a case so that you understand exactly how we use it in practice. This is one of the cases that's presented at the Undiagnosed Disease Network, where a lab, Michael Wengler's lab at the Texas Children's, was handling. And this is a 13-year-old female that presented with intellectual disabilities and some mobility issues, and also some facial dysmorphic. Oftentimes you have case equals to 1, and that is a big challenge for a lot of machine learning programs, because you don't really have, until you figure out what's going on with this case, it's very hard to identify another case that you can say, oh, they are in the same label. So the way to do that is we run the algorithm using AI Marvel, and then get a short list of candidates, like 3 genes, 4 genes, and to figure out what's potential candidates here. And then you move that short list to the fly core, where the fly geneticist will try to kind of further validate which one of this is really, really relevant to the particular patient. But there's a huge evolutionary jump from human to fly, and the first thing is, will it work at all, right? But I hope you can agree that, like in the first page, we already done this repeatedly for many, many cases, probably 71 cases. And the way to do that is actually to humanize the fly. In other words, this is, we call it the Trojan horse in the fly geneticist. You kind of hijacked the endogenous locus of the gene, if the gene is evolutionarily conserved. And then you try to remove the fly gene and replace it with the human gene. And the first thing you want to make sure is that the human gene will function similarly in the fly, at least not causing a lot of problems. And here you can see that the first column is you look at it carefully about the phenotypes. In this case, we look at the wing morphology. The first column is the wing of a fly, and the second column is the humanized fly. In other words, when you replace this gene called MRTFB, so this gene can actually function mostly normally in the fly. There's a tiny, you see this arrow, which is a tiny kind of connection there. That's kind of not closing as compared to the healthy fly. But overall, it is still there. So like the majority function is there. What happens next is really the key. What you do is you express, instead of expressing the reference gene, you actually bring in the human mutation, the patient, particularly the patient's mutation in, and see whether it's actually causing any problem. And the third column is actually when you kind of particularly introduce the point mutation that a patient has, you can see it completely destroyed the wing formation. Right, and that's where you do the same thing for the other genes and you see nothing. There, of course, then you will say out of the three, I think this one is the most probable one because you clearly see it destroyed the kind of the formation. There's a developmental process being disrupted. Of course, you do not want to draw the morphology phenotypes of the wing to the facial phenotypes. That will be a very long stretch to call these are related, but there are fundamental processes that are conserved. Once you have one case and you say, oh, this girl has a mutation in MRTFB, what you can do is to go to Gene Matcher. There are many such kind of consortium. You can say, I have a patient that has intellectual disabilities, has a facial dysmorphic, and I think this is the gene, this is the mutation. You send it through an email blast, and then trying to hope maybe somewhere in this world, another clinician has a similar case, and they can look at your mutation and say, wait a second, I have another patient in my clinic that has similar phenotype, and the gene also has a mutation in the position. And luckily, we have another case actually in a totally independent, unrelated family. As you can see, the mutation is in a different place. This one is R in position 104, and this kid has a mutation in 91. But they're close enough actually in this RPL protein domain. It's basically an important function. They're proximally close to each other. And once you have multiple patients, similar phenotypes, similar mutation in the same gene, then you can call this as a kind of a solved case because you're pretty sure at that point that this is probably the causal genes, right? And of course, you can see that when we express the second mutation, A91P, the fly also has very similar phenotypes. The wing is destroyed. So this level of consistency is where you can confidently deliver this back to your patient and say, we probably found a cause for you. Now you move to the next phase and finding a therapy. And I think many of the speakers yesterday have shown the promise of gene therapy. And in particular for the rare diseases, the first step is to figure out which gene is causing the problem. Then you can start this therapy development process. Well, although fly is a very powerful genetic tools, right? And for one of these genes, you probably take 6 months to a year to figure it out. But remember, you have multiple candidates, you have multiple patients. When you scale it up, it becomes a problem. So these modern AI tools actually can help here because genes to the cells are the words to our languages, to the sentences. And there's actually many papers nowadays kind of building this gene expression large language models where they're taking the single cell expression profiles. In this case, we're interested in fly. So we took 2.5 to a million fly cells across different cell types, and then you tokenize the expression into the gene names and the expression levels, literally making it a sentence, and then you mask some of them, you hopefully encode, decode it, and will be able to predict what you masked, right? This is kind of a typical training process, a pre-training process that many of the speakers throughout the symposium have discussed about, and we have basically the similar self-supervised learning phase, pre-training phase, and then you have this, for fly particularly, unlike human cells, there is a very good study about the development, the cell lineages. We know many cells, how they come from embryos to all the way differentiating into particular neurons. And here, basically, we have a hierarchical lineage about cells that will enable us to get a better understanding of their function. And then once you have the pre-trained models, and then you can basically have downstream tasks, whether it's cell clustering, other things. Cell types in particular, I think the crown jewel is the in silico perturbation, because that's where it is relevant to disease gene prioritization. You want, instead of physically perturbing the gene, you want to in silico perturb the gene and see, is it going to have a notched wing? Is it going to have a, right? And then that is basically you in silico remove or overexpress a gene and see what is the delta change in this disease status. And hopefully you have these strong outliers effects that you can kind of move down to your downstream analysis. And of course, you take patients' VCF out, there's still a gap between the mutation, actual DNA sequences versus genes, and there are many protein language models we can use to embed these changes, and hopefully that we can kind of make a prediction there. And lastly, I'm not going to talk about how gene therapy or antisense oligotherapy is going to develop, but that's like a huge topic, probably deserve another time. But if you have these therapies, you really need clinical trials to really figure out what's going on here. And being in the Texas Children's Hospital, one of the largest pediatric hospitals in the United States, we have any given time, we have probably 200 plus trials going on. And every single trial, there's something has to be done first, which is the cohort building. Every trial requires a cohort that has many questions that are actually specific to the patients, and collect information from the operating system of the hospital. Sometimes you use a Cerner, sometimes you use Epic. Regardless, like you have a separate registry called a patient registry. There are a lot of studies about this, like most of the time, like probably half the day of a physician's life is spent on documentations. And then when you spend 50% of your working out 10 hours shift, documenting, you probably have less time to kind of really take care of your patient and thinking what's going on with this particular case, right? And this is kind of a major pain point of the hospital operation. And how can we solve it? Because this cannot be, many of the tools I've mentioned about lives in the lab, lives in the research lab, a major kind of NIH funded project, but this is different. This has to live in the real world environment in the hospital. So it requires different skill sets. The EHR is a kind of transactional system. What it's really good at is to get reimbursement from insurance companies, and transactional so that we are legally compliant as a hospital. It's not for clinical trials. It's not for 360 view of your patients. How do we do that? What if you have a registry and the registry can fill itself, right? That's basically the big goal here. And to do that, we have to collaborate with industry, because we don't want to build a system when you have like 4 million patients transactions every year, and then you have to search the system, and you need a certain speed and stability of this. So we worked with the industries to figure out how we can really elevate at this point. In this case, we typically use a red cap of the registry, and then typical process is there will be a research coordinator by the physician. They'll go into the system line by line, look at the notes, look at the records, pull the information. A typical registry can have 1,000 questions per patient, and then they have to basically spend hours of time for every patient to fill that. And this is a perfect job actually for large language models, because I think I don't have to convince you guys there are multiple examples throughout the symposium where you can give a document and do semantic retrieval, and there's a pretty good high accuracy nowadays in terms of getting the right information. But I want to highlight one point, particular for example, this is FPIES, which is a very rare food allergy disease, right? There are certain definitions as like a chronic allergy and acute allergy. That's different than what the large language model will understand, because they learn it from the internet. But when you're dealing with a small niche, like a rare disease, those chronic and acute allergies has a completely definition. The physician has to go back and then fill in the exact requirement to instruct the large language models to pull the right information. We did the first run, it was pulling wrong information. And when the doctors kind of revise the prompt and give an examples, then the retrieval becomes much, much more accurate. So basically, what we did, we gave the LLM to select the patient questionnaires and let it kind of do the agent searches to answer those questions. And then the agent will also pull the supporting evidence from Epic system to say, this is where the notes, this is the day of the transaction, and can you approve, do you agree or disagree? Then research coordinator will basically review this evidence and then push it back to the registry if they agree or make modifications. Under the hood, there are three components: the data layer, the agent layer, and the application layers. And because we're really deploying this through our system, so we basically build a backbone on the Foundry system. One thing that is they're really good at, I feel like, is the ontologies on particular healthcare data, and also their speed. Whenever you're searching a large amount of record, it's done very quickly as compared to traditional database like in Epic. So here we did several pilot studies, and we're currently rolling it out through our hospital, where in the manual registry, in the FPIES case I mentioned earlier, the food allergy case, severe food allergy case, typically a research coordinator will take 60 minutes to go over probably 1,000 questions, and then if you have 100 patients, you scale it up linearly. There's really no way to kind of cut short the time. And then the entry, basically the main job of the coordinator is data entry and the manual navigation, so much less review. However, if you let the agent model to do this, most of the time the poll is actually doing the background. Most of the time is spent at reviewing the record. There's a huge reduction in terms of the time spent per case. Hopefully, I want to close. You can see that how we put together this loop, starting from the patients, using AI to narrow down, validation using FLY and other human genetic evidence, pushing it back to our clinical trials, and then every step the AI is facilitating the process, sometimes even playing a major role in selecting the direction to go in terms of which gene and variant to study. And with that, I would like to thank my talented team and my great collaborators, and really this is the entire Texas Children's team that's solving these cases over the past 15 years.
Thank you. We have time for questions.
Hi, nice talk. I have a question about how to treat this unfortunate case. As a neurodevelopmental disease, and these are kind of a gain of toxicity genes, so the neurons has already wired, and like how you identify the gene and how do you develop treatment?
So there are loss of functions, there are gain of functions in terms of this mutation, and sometimes damage is done and the kid's already past the developmental stage, and it's going to be hard. But there are cases where, for example, the lysosomal storage like diseases where you can still reduce the toxicity because it's a kind of a garbage accumulation of some toxic proteins. If you the therapies that, for example, the ASO antisense oligotherapy can still kind of reduce the amount of a toxicity protein buildup, and of course it's not going to cure the disease or it slow down the course and maybe sometimes reverse the course of the disease progression, and that's how we're doing antisense oligos, and they're also kind of a CRISPR.
I was wondering if you've thought of expanding this to, I mean, I have personal, and several people in the room have a personal connection to epilepsy through our practice. We call epilepsy a disease, but it's really a constellation of many, many, many diseases, and they have a similar set of endpoint phenotypes. Have you looked at that? Because you do have many individuals who've had tests with genetic variants identified, but those genetic variants on an individual by individual basis are rare in and of themselves.
Right. So we have another project. It's called a Biobank Project at AIAS Children's, which the epilepsy is, as you put it very nicely, that is really a constellation of diseases. Many of them have a foundation, genetic mutation foundation there. Many of them actually we don't know what causing them. And there are labs at the TCH. Usually they're using mice to model that seizure process, and fly is another example. So we do have a kind of a project. Every single severe patients passing through AIAS Children's will be sequenced, and their genome will be analyzed. And the power relies on if two seizure patients have very similar phenotypes and they have the same mutation in the same gene, then we can kind of quickly narrow it down and push it for kind of a functional studies.
I'd like to take Chairman's prerogative and ask you a genetics-based question. So, as you know, once you have a phenotype in a fly, it can be very powerful for doing enhancer and suppressor screens to find interacting genes, which can tell you a lot about genetic background effects. Those are very laborious and time-consuming. So, have you explored using AI to narrow down the space of potential interacting genes through gene interaction networks and in silico screens, and then try to see if you can identify those genes and find their human homologs?
Yes, there's an effort. Juan Bota's lab particularly does that. The modifiers, enhancer and repressors. There are two approaches actually. We have robotics that can speed up the screening process, which is labor intensive, right? So every step, for example, to figuring out, in fly genetics, you need the virgin females, but traditionally you have to use an eyes to kind of a technician will be there to sort out these flies, but now there are machines using computer vision to quickly identify, oh, this is a female, this is a male, and then like every step there's a process, and the climbing assays, there are robotic computer visions to automatically calculate which genotype, right? And then on top of that, once you accumulate enough data, then you can extrapolate. You train these models, say, oh, I have a Alzheimer model, I have like a genetic model, like MTRF-B1, and then can I use these foundation models and to figure out which one is a repressor, which one is an enhancer. The key, I think one challenge that compared to other AI field is that oftentimes you have the data and you're sure this is the data, a computer vision problem. And in our field, oftentimes our job is to find out what is the data to make that prediction. For example, what information is really predictive of repressor status? It could be gene expression, it could be the DNA sequences, could be the gene network, and also of course the data we accumulate in the lab. So that's a big challenge for us to find out what data is really usable.
Thank you. Other questions?
I have a very early question to your whole method. You seem to be doing variant identification in the genes, and
Right.
you started with whole genome sequencing, it sounded like, and then maybe went to exome sequencing. I'm not quite sure what the distance was, and then it seemed like you went to single gene variant analysis. How does that work in this type of environment, which might be multi-gene based? I just have a very basic question at the beginning, which is you're in variant analysis, maybe scato-like analysis. I just can't figure out how do you bridge if it's multi-gene?
Right, right. So apologize for not being clear in the in the introduction part. So oftentimes you think about complex diseases like diabetes and Parkinson's and Alzheimer's. These have multiple gene contributions, and then the effect size is oftentimes small unless it's a familial case, right? So those will not fall into today's talk. The rare diseases that we deal with oftentimes has very high penetrance, and then they have low prevalence, of course, in the population. But most of them are monogenic. There are some digenic, and we basically primarily focus on whole genome, whole exome.
Okay, I think in the interest of time, we should move on. Let's thank Dr. Liu again. So our final speaker today is Tom Miller. Tom was a professor at Caltech, my own institution, for 10 years, and left to found a startup company, Iambic Therapeutics, which is focused on finding new drug development candidates for treating various diseases like breast, lung, and brain cancers. He received his PhD in theoretical chemistry from the University of Oxford, and his title is on the slide here, so I won't read it. Tom.
Thank you, David. It's wonderful to participate in this symposium, and many thanks to the other speakers. I'm going to be talking about a slightly different aspect of the challenge, the healthcare challenge. Specifically, once we identify the target of interest, how are we actually going to find the medicine? How are we going to use AI to explore the possibility of new medicines and to deliver those successful new medicines actually to patients? And I'll be discussing how we do that at Iambic. I'll start with just a little bit of a reminder of the journey in going from a target of interest to an actual clinical candidate. And that's sort of summarized on the top slide here. Once you have a particular target of interest, I'm actually trying to figure out if I have a laser pointer. I don't think I do have a laser pointer. But anyways, if you go along the top in terms of the actual journey that a molecule has to take, you might have a target of interest. Then chemically, you'll try to identify initial chemical matter that might be a hit that would start to engage that target. Then you would start to ask whether or not you're selective for that target versus other things that might cause toxicity. Then you might start to ask about whether or not you have the right metabolic properties so that it'll actually stay in the blood system and whether it gets to the right tissue. Then you'll ask whether or not it actually can be developed at scale and have the right chemical properties to actually be manufactured into a drug. And then you have to take it into ever higher species, like into rats and then dogs and then to monkeys, before you can actually safely take that into humans, all of which entail different stage gates and different risks. So the journey, even before you start doing clinical trials, is a multi-parameter challenge of really trying to identify the right molecules that can satisfy all of these different stage gates. And you can really imagine what happens. If you solve for the first thing and don't look at the second thing, then you have a probability of failure at each one of these different stage gates, and that leads to the extraordinary failure rate of typical drug discovery and the associated long time scales. What we seek to do, and what others are also seeking to do, of course, is to try to take all of that available data from all of these different stage gates and to try to fold that into large foundation models that allow us, from the earliest days of a program, to predict across all of these different considerations, and therefore to ultimately try to improve the probability of reaching clinic and to doing so in a timely and successful way. I thought the early discussion from Christopher Bishop about the fifth generation of science, is very apropos for the way in which we are really approaching that. We will use AI to drive what experiments that we're actually going to do. We'll use those experimental data in a feedback loop to inform the next generation of the AI, and we drive forward on that on a rapid time scale. At Iambic, we take all of that external data and we build large foundation models. The two flagship foundation models that we utilize at Iambic that we've developed are one called Neuroplexer. We actually heard about Neuroplexer when Zhiran Xiao, the prize winner, described that yesterday when he was working at Iambic in my group at Caltech. And then also Enchant, which predicts on basically a whole battery of both preclinical and clinical stage properties. So in a sense, Neuroplexer is how we predict the three-dimensional structure, how the molecule will bind in 3 dimensions to a protein of interest. And Enchant, which I'll actually dig into somewhat more in this presentation, allows us to really predict across all of these other different properties. What we then do is we combine those AI designs and we implement them in terms of plate-based chemistry, automated chemistry that are executed using robotic liquid handling. We then take that through to plate-based biology across dozens of different assays in-house. And we'll do that for thousands of new molecules each and every week. So we'll go through new designs, we'll make the molecules, we'll ensure that they can be synthesized with high probability, we'll then assay those molecules, we'll automatically retrain the models, and we'll then iterate in a closed loop cycle, again, for thousands of molecules every week to try to drive forward in terms of a discovery process. There's a lot that's summarized here. This is a big thing. Iambic is now a company that's over 100 people that are really executing on all aspects of this on a weekly cycle. I want to dig into Enchant, the way in which we use AI to really predict the dozens of different properties that are necessary for bringing a molecule forward to clinic. And really Enchant rests upon 3 different concepts. The first concept is that it has to be flexible in terms of the data sources that it can use. As I kind of emphasized on the previous slide, there's not just one kind of data. It's not just binding data, or it's not just PK data, or it's not just toxicity data. There's lots of different data that you want to take advantage of. So what we want to be able to do is, as you can see on the left-hand side, make sure that we build a single framework that can flexibly take not just data sources, but actually different data shapes, and to fold that into a single learning framework. So Enchant achieves that. I think another departure for Enchant versus more typical deep learning approaches in many drug discovery activities is illustrated in the middle panel there. Oftentimes, if you want to predict on, say, a binding affinity for a target, you build an AI model for that. Or if you want to predict on the solubility of your molecule, you'd build a model for that. Or if you want to predict on the metabolic properties, you'd build a model for that. You'd build different models for each thing. And on the one hand, sometimes you don't have enough data to have a good model, so that's one limiting factor, so you'll have poor predictions. And the other thing is you don't benefit across the mutual learning of those different properties. So but by the traditional approach of building different models for each different task, you're leaving a lot of opportunity on the table in terms of realizing data scale. And that's what Enchant definitely strives to do. And then the third thing, why that's really exciting, is kind of illustrated in terms of a paper from Google on the right-hand side. Typically in drug discovery, what you are faced with is the fact that no individual data set is big enough to really reach the size scales that are necessary for seeing the exciting emergent behavior of transformer models. So for example, if you even took the entire PDB, it's just not enough data to predict the structure of proteins using a transformer approach, and therefore more specific approaches are needed for that task. But if you can get to the right data scales, you can start to see the exciting emergent behavior that we're now familiar with ChatGPT and with other large transformer models. And that's illustrated here on the right-hand side. What they were doing in this particular example is they were doing question and answer in a language context. So they were basically asking questions in Farsi, and they were training an AI model on more and more data that was fully excluding Farsi. Right? So this was a multiple choice question that they were basically asking. Presumably there were 4 answers you can see on the right-hand side, so random guess basically gives you a 25% response rate. And then they increased the size. And what they found is that at a particular model scale, they started to see that there was a huge uptick in the ability to answer questions in Farsi, even though seemingly the model had been presented with no Farsi in the first place. And that's not quite true. There is a little bit of Farsi that the model had been presented with. It was presented with the question, which was posed in Farsi. So the exciting thing is, if you have a sufficiently big model, then even a small amount of data, like the amount of Farsi in the question, is enough to give you the learning capability to be able to begin to answer questions in that domain for which you otherwise had no data. This was really exciting to us because this is exactly what happens in drug discovery. When you're trying to predict on human clinical behavior or human clinical output, you don't have much data. You're always in an extreme data poor regime. There's some data that you can manufacture at scale, like cellular data, like small animal model data, but you can't manufacture at scale human clinical data. So by being able to have a framework where you could reach the model scale associated with those easily manufactured data points associated with lower biological tests, and be able to transfer based on very small amounts of data to the higher value regime, namely the human clinical readouts, this signifies that an approach of this kind can actually achieve that. And that's exactly what we see in the approach that we're using, which I'll show you. So the way Enchant works is it's basically a transformer model. It utilizes biological data of many different forms. It actually uses 15 different distinct structures of data. We do have a paper that was published in ICML. We basically include molecular and biomolecular structure data, all sorts of sequence genomics data, any kind of tabular assay data that you would like to use, physics-based data like the quantum mechanical data that Christopher Bishop was talking about, as well as unstructured biomedical text. And that gets rolled into a very large overall transformer model. And it starts to exhibit the emergent behavior that it was anticipated in that paper from Google. And I'll walk you through this learning curve that illustrates the emergence of that. So what we're doing here is we're basically predicting, the y-axis is a measure of accuracy. What we're showing is the degree to which we can take a new molecule and predict the degree to which it is metabolized by a human being, basically the oral bioavailability in a human being. So this is one of the key things that dictates whether or not when you take a pill, it's actually going to be in your bloodstream at the right level, and it's one of the two major reasons you would fail, for example, a phase 1 clinical trial. Fine. So the y-axis is basically being able to predict that important human property. The x-axis is a learning curve. So it's increased in the amount of data that we're including in that. But we're doing that in a very specific way. We're using only a tiny amount of clinical data. We're using like 20 data points for randomly chosen drugs. So 5% of the total available clinical training data. Very sparse. And what we're increasing on the x-axis is more and more preclinical data of the kind that you can easily manufacture in a laboratory setting. More cellular assays, more mouse experiments, that kind of thing. So we're increasing that on just different drugs, increasing the model scale, and what we're doing is we're holding fixed the amount of actual clinical data. And what you can see, very excitingly, is that indeed it learns. Even though it's not getting more clinical data, it gets better at predicting the human clinical property by training on more preclinical, easily manufactured data for other molecules. So that's exactly the thesis that we wanted to see. And what gets particularly exciting is what we see on the right hand when we start to benchmark the accuracy that we're able to get in comparison to models that were specifically trained using 100% of the clinical data, not just 5% of it, 100% of the clinical data on that specific property. So what you can see is that with 5% of the data, Enchant is able to surpass the state of the art in terms of models that are using 100% of the data on the properties of interest. And of course, when we then don't encumber it that way and we use the full amount of available training data, it further increases and actually sets a new standard, which is extremely high predictive accuracy, almost nearly 0.8. And that's a big deal because basically that gets us to within roughly twofold prediction accuracy for this property of interest. And why does that matter? Because that's how drug discovery really gets accelerated. The normal way in which you try to find out what the human clinical PK is going to be, what the bioavailability of your drug is going to be, is using the process at the top. You have to make the molecule. You have to synthesize it. You have to do in vitro assays. You then have to do in vivo assays. You then have to go through actually a very elaborate extrapolation process. And then, after about 6 months, where you basically have done all the work, only then do you know within a factor of 2 whether or not this thing is actually going to be exposed in the human body at the concentration that you want. What I just showed you on the previous slide, and what we find, is that ENCHANT is allowed to do that and achieves that same predictive accuracy, but you don't have to make the molecule. You don't have to do any of those intermediate steps. It allows you, when you are at the earliest stages of your drug discovery program, before you commit to the scaffold, before you commit to all of your lead optimization, you know that you're in a family of molecules that are going to be de-risked with respect to your phase 1 property of whether or not you get that bioavailability. That's why this actually matters. Great. We use this not just on human PK, we use this across many properties. I'll show you actually an incomplete list on the next one. But what I really want to show here is that ENCHANT is built from the ground up. It's trained from scratch on biomedical data. And that is one way to reach the necessary model scales, by taking all of that multimodal data and to lump all that together into one big transformer model. You might ask, and we often get asked, whether or not you could have reached the model scale using the more conventional large language way and then just fine-tuned it for drug discovery. Why not just take ChatGPT and then fine-tune it for drug discovery? Would that work as well? Well, in fact, that experiment was actually done. That's what TXGemma is, that was developed by Google DeepMind. And what they did is they took the Gemini large language model, which reaches model scale not on biological data, but reaches model scale based on human text, and then fine-tuned it for drug discovery. And what you can really see is across a whole host of different relevant drug discovery properties that were tested, ENCHANT is vastly more accurate than TXGemma is, kind of illustrating the importance of building up in the way that we did. We use this broadly across every single property of interest at Iambic. So these are actual just different properties that we are using today across all of our different drug programs, where ENCHANT is allowing us to predict across all of these different things simultaneously. It includes target-specific properties, whether or not we get activity on different targets of interest, physical chemical properties, whether or not it's soluble, whether or not it's in the right sort of blood models, whether or not it has thermodynamic solubility, whether or not it has the right acidity, whether or not it gets across the lipid membrane, both in vivo and in vitro pharmacokinetic properties, including across different species and in human, as well as a ton of different safety properties as well that you have to de-risk. Is it going to have GI tox? Is it going to have different other sources of heart-related tox, et cetera? So we broadly use this as a single framework for predicting all of those different things and benefiting from the same sort of aspects that I was illustrating in the specific case of the human PK. And what we do is we use this to actually try to find new drugs. And we use this to try to find new drugs, and we focus on cancer for our 3 lead programs. So we have one program that's in clinic. I'll actually tell you a little bit about it. We have a second program that will be entering clinic very shortly, and well, we have 2 additional ones that will basically be in clinic in the next 6 months. And actually, with neurology relevance, we're also working with Lundbeck on a major migraine target to try to use the same framework for addressing that important unmet need. I want to tell you about our most advanced program, which is a HER2 program. HER2 is an oncogene that's very well known, but it's a huge unmet need in terms of many different forms of cancer. HER2 mutations or overexpressions or amplifications, all 3 different distinct kind of molecular modalities, will lead to a variety of different forms of cancer, including breast, lung, gastric, colorectal, et cetera. There's a large number of people that are impacted by this, and it's really important to know that one of the really devastating aspects of HER2-driven cancers is that they will metastasize to the brain. So over 40% of breast and lung cancer patients will actually find that the tumor transfers to the safe haven of the brain, and that ultimately drives morbidity. So that informs what we wanted to design. we wanted to design a molecule that could hit the mutations as well as the wild type forms of HER2 to address both the mutations and the overexpression and the amplification. We wanted to make sure that it had brain penetrance so that we could address these brain metastases. And furthermore, we wanted to make sure that it was able to hit the HER2 target hard enough without incurring the typical dose-limiting toxicities of severe rash and severe liver tox, which are associated with EGFR and which have been the primary limitations of other drugs in this space. So those are the three things we wanted to get right. Pan-mutant, brain penetrance, and got to be highly selective for HER2 versus the extremely similar binding site of EGFR, which drives the tox. We are now in our phase 1 clinical trials. I'll talk to you about that shortly. We actually just last week reported our first clinical data from the phase 1b trial, so I can actually now show you that in this public setting, which is exciting. We're open for this particular trial in over 28 different sites currently, and it's expanding up to 60 over the course of the next year, both in the US and in Europe and in a few Asian countries as well. The platform was really important for how we actually discovered this molecule. Kind of using the closed loop cycle that I talked about at the beginning, we drove on 20 different properties, including the on-target activity, avoiding the off-target activity, all the metabolic and PK properties, the brain penetrance. We executed over 10 closed loop cycles in the first 8 months of that program, and it unlocked a ton of chemical space. Blue dots are previously known molecules in the literature. Gold dots are new compounds at Iambic that have, as you can see, very differentiated selectivity and overall ADME-PK properties. And then to understand the pan-mutant activity, we used our structure-based approaches as well to really find the ultimate development candidate. Just at a very high level, how this stacks up over what had previously been developed. It's just much more selective. You can see in those right two columns that over 1,000-fold selectivity allows for you to hit the HER2 target while avoiding that off-target EGFR tox. It's active across all the different mutations, and it's highly brain penetrant. So it's really different from any other drug that has previously been discovered in this space. Only one of these is even approved, the tucatinib drug from Pfizer. The others are actually earlier stage experimental drugs. And you can see how IM1363 is differentiated on all three of those different key axes that matter for patients. And this is just the data. This is how it actually is working in people. So I'll skip 2 years worth of animal data, and I'll just show you the people, and that's what we're actually showing right here. So what we've been able to do is show for the first 18 patients that have been treated with this. There's actually over 36, but only 18 of those are actually at the necessary dose levels because there was a dose escalation process. We're really excited by the breadth of activity that we're seeing as well as the safety profile that we're seeing. I won't walk you through entirely, but what you're seeing here on the right-hand side, and I'm sorry for the smaller text, is basically each bar is a different person, and it's their tumor. It's the largest, or it's the combination measure of all of their tumors in terms of that tumor size. And what you can see is that basically over 80% of patients that were dosed at the relevant dose levels are saying that the tumor either ceases to grow or is substantially reduced. And I really want to emphasize that these patients are extremely heavily pretreated. In some cases, this particular line shows the number of lines of prior therapy. Some of them have had 6 prior lines of therapy, or 4 lines of prior therapy, or 5 lines of prior therapy. You can see that. And you can also see that the tumor types that we're dosing here are extremely diverse. One is a lung cancer patient with amplification. One is an ovarian cancer patient, just going from the right-hand side. One's a breast cancer patient. That's actually their brain metastasis that reduced there. One is a lung cancer mutation patient. One is a gastric cancer patient. One is a renal cancer mutation patient, for which there's actually no approved therapy right now. So that's just even kind of starting from the right-hand side. So we're very excited, even in this early data, to see an extremely broadly active drug as well as an extremely well-tolerated drug, because in contrast with other HER2-directed drugs that have been developed, which are dose-limited by both a severe rash and severe liver toxicity, we see no evidence of that to date, actually, in this drug. So very encouraging on that count as well. So we're going to keep pushing that forward, and we'll have additional data coming out in the upcoming year as we push towards our first approval. You know, in the popular literature, and this is my last slide, in the popular literature, you know, you're starting to hear a lot of questions like, where are all the AI-discovered drugs, right? You know, in some sense, we've been hearing about how AI should be helping healthcare and should be designing drugs. And I think, you know, really, that's the right question to ask. And we are excited in the measurable way in which this approach is actually bringing medicines to patients faster. So what you can see here is an analysis from the Boston Consulting Group of the fastest examples of AI-driven drugs. Typically, it takes about 6 years to get from a program start to clinic. In this program, we did that in less than 2 years, and now we're at that first asterisk where not only are we in clinic, but we have actually shown that we have a safe and active drug with proof of concept data. And you can even see with that second asterisk there, that's the timeline, which is roughly in 2028 for our first potential approval of this drug. So we're excited to see that kind of come together and that fruition. So yeah, I'll just leave it with that. I'm happy to take any questions. Basically, AI models plus laboratory automation pointed at important unmet need for patients can ultimately lead to what we hope is better outcomes. Thanks.
Thank you. Thanks, Tom. Great talk. We have time for some questions.
You particularly indicated how effective to predict PK. I wonder, what about the efficacy? Was there any like a strong signal you can see even in the early discovery stage?
Yeah, absolutely. So when I showed, I kind of did that deep dive around the specific property of PK, but when I showed you kind of a list of like 40 or 50 different properties, about 10 of those were specific things that are predicting on the activity and the efficacy of the drug. So yeah, that's absolutely something that we're also able to predict and use the model to predict as well.
Hi, I have a question from the point of view as you are a CEO of a startup company. So from some of my sources, analysis of Wall Street, including BOA and Morgan Stanley, they still hold a point of view kind of conservative on AI discovery drugs. So what do you interpret the gaps between Wall Street, the capitalism world, and the moving forward of true AI discovery drug?
Come to San Francisco, I get a VC question. It's good. So no, it's great to know.
Wall
But
Street.
you know, look, you know, I think that we are a second generation AI company, right? We are not, you know, I think a lot of people were able to found initial companies on a lot of promise for AI and hope, and we have always focused on execution. So when we talk to Wall Street, when we talk to investors, yeah, we explain the way in which AI is helping us, but the proof of the pudding's in the eating. I mean, we point to the medicines that we're actually able to make. We point to the clinical candidates that are created. So many of our investors at Iambic don't care about AI. They care about medicines for patients, and they like that we have a platform that allows us to do that, and that's core to how we create value.
I had a question about models for predicting PK. Do you know how well you can predict PK if you exclude data from in vivo studies, just using in vitro and other data?
Yeah, we can cut the data in different ways, and as you you know, go further from, as you have to translate further, as you can imagine from any machine learning context, you will see a weaker correlation. I think the important point about that slide is that I'm not doing an extrapolation. I'm not saying you have your molecule and now you have in vitro data and then you have in vivo data and I'm just going to predict the last thing. What I'm showing in that particular slide is that I have 600 other molecules that are across all diseases and have nothing to do with my chemical series. And if I start putting in data for those molecules preclinically, I can predict for my molecule, the one I care about, actually in terms of clinical. So that's the particular point that we're making there.
Yeah, and thanks for a very nice talk. So I had a question about delivery systems. There are a lot of systems in the market like liposome, nanoparticles, and the idea is to cross the blood-brain barrier. So the compound you had has very low brain penetration.
Yeah.
Exosomes can cross the blood-brain barrier.
Yeah.
Now, can the system tell us that how to design an exosome delivery system whether we can target the pancreatic tumors or brain cancer tumors so that we can modulate it in a preclinical phase and then have more success on the clinical side.
Yeah, that's great. You know, Iambic, in principle, you can take frameworks like this and point them at lots of interesting questions and lots of specific strategies, like you're actually outlining. You know, the primary mode that we focus on is basically oral drugs. So you take a pill, it goes in your bloodstream, and we do optimize the molecules to get across the blood-brain barrier, as well as to target tumor tissue. So that's the other way in which we do that. But you could imagine and employ very similar models to try to get to other tissue-specific outcomes, or to even query whether different delivery mechanisms are going to be more or less effective. It's not the primary thing we do, but you could absolutely take this approach.
Yeah, so great. So thank you, Professor, now the CEO. For the HER2 is a very well-known target in biology, and we have a lot of antibodies already target against that. Now, small molecule versus antibody, the specificity, have you done any comparison with that, the biologics target? And also the new trend in drug discovery is the antibody-drug conjugate. Do you have something in mind in that?
Yeah, no, that's exactly right. And of course, so yeah, the bottom line is there are some advantages of small molecules versus antibodies. Like, for example, getting across the blood-brain barrier is a major advantage. But we really don't see it as an either or. Small molecules are a really natural combination partner for antibodies and for ADCs. And in fact, in breast cancer, we just announced last week that we are opening 2 different second-line breast cancer cohorts where we are combining with 2 different leading antibodies, Herceptin, as well as a novel one that Jazz Pharmaceuticals is actually giving to us as a next-generation one. So we don't see it as an either or. We see it as, you know, almost all cancer therapies are given in combination. What you want are unique modalities that can really hit the target hard and be tolerated to where they can actually be safely combined. So small molecules are very important in that overall approach.
Okay, it's time for lunch. I think we should first thank all the speakers for an excellent session. And thanks to all of you for participating and asking questions.